Hi, I'm Jose Blanca and this is the course about population genetics. Today we are going to talk about population characterization. We are going to start with allelic and genotypic frequencies. First of all, what do we mean by allele? There are two different meanings that we use for alleles. Allele is a form of a marker, like for instance a SNP, or in general a locus in a population. We can say things like in the Spanish populations there are 3 alleles for this locus, or there are 3 alleles for this marker. This is one of the meanings, but there is another meaning, and it's the set of particular markers, the particular alleles that an individual has. For instance, we can say this individual from the Spanish population has 2 alleles, and both alleles are the same because it's an homozygous individual. In the first meaning, what we are talking is all the forms present for a marker or a sequence or a haplotype, in general a locus in a population. In the second meaning, we are talking about for a particular individual which are the forms present in that individual, and these forms can be the same if the individual is homozygous. In general, the nomenclature in genetics is quite confusing. That is why I like to clarify a little bit the nomenclature before we start. So we have 2 meanings for allele, and there's also another complication, and it is that it is quite frequent to use the term gene to refer to these meanings. For instance, we can hear things like, especially in breeding, like we have a gene for resistance, when in fact most of the time in that case what we mean is that we have an allele, the resistant allele for the locus. Okay, we have already said what is an allele. One important, very important idea in population genetics is the allelic frequency. Imagine that you have one locus with 2 alleles, capital and lower A. So the frequencies would be how many alleles of each kind of alleles do we have in the population. For instance, how many capital A alleles do we have divided by the total number of alleles. That would be the frequency of the capital A allele. Usually when we have in population genetics 2 alleles, we talk about P and Q. P is the frequency of the capital allele, and Q is the frequency of the lowercase allele. And of course, the sum of P and Q should be 1, because every allele should be either capital A or lower A, because we have only 2 alleles in the population, as we have said. So the sum of the allele frequencies in general should be always 1. Another concept that is you will hear in population genetics is the major and minor allele frequencies, MAF. It's a little bit confusing because sometimes when we talk about MAF, we talk about the major allele frequency and sometimes about the minor allele frequency. If we only have 2 alleles, the major allele frequency would be the allele with the highest allelic frequency, and the minor allele frequency would be the allele with the lowest allele frequency. So in this example, for instance, we have a major allele frequency, it would be capital A, because it has a frequency that it's 0.8, that it's higher than 0.2, this would be lower A, would be the minor allele because it has a lower frequency. If the locus is not biallelic, then it is a bit more confusing, because if we use the term minor, in this case, what we are referring to? To the allele with the minor frequency. Yeah, but we have several alleles. So in that case, the term that is well defined is major allele frequency. That would be the allele with the major, with the highest allele frequency. While the minor allele frequency, it's not that clear. What do we mean by that? Because usually, when we have more than two alleles, what people talk about when people talk about minor allele frequency, it's the sum of all the alleles that are not the major allele frequency. Okay, that is equivalent to convert our marker, our loci, our locus, to two alleles. The major one and all the other ones together would be the other allele. Okay, if we have, for instance, three minor alleles, A1, A2, and A3, we sum them up and we get only one allele. Okay, that is the sense in which people usually talk about major and minor allele when we have more than two alleles. When we have calculated the allele frequencies for many when we have calculated the major or minor allele frequencies for many loci in the population, like for instance thousands of SNP markers, we can create an histogram of the allele frequencies. How many markers do we have in which the minor or the major allele frequencies are within a bin? For instance, how many SNPs do we have that have an allele frequency between 0 and 0.01? How many between 0.01 and 0.02, and so on and so forth? So we can calculate this distribution, and this distribution is called allele frequency spectrum, or sometimes is also referred as site frequency spectrum. Okay, this is a distribution that characterizes quite well the allele frequencies in a population, because we have the distribution for many markers. As you can see, most loci, or if we have markers, for instance, most SNPs in a population will have a very low frequency for the minor allele. So these markers will be almost fixed. This is not a characteristic of this particular population that I have represented here. This is a very general phenomenon. Always in a population, if you calculate the allele frequency for a random SNP, for instance, the most likely thing that will happen is that that allele frequency will be very, very small for the minor allele. So most individuals in the population will be homozygous for the major allele, and very few individuals in the population will have the minor allele, usually in heterozygosis, as we will see. So this is a general feature of the populations. Okay, most loci that are variable are almost fixed. The minor allele is present in very few individuals. The reason why it's because there's an equilibrium between mutation and drift, and we will see how this equilibrium leads to the creation of this shape for the allele frequency spectrum, for the distribution of allele frequencies. I've talked about fixed, variable, and polymorphic loci. Okay, what's the meaning of that? Fixed means that this locus is fixed. A locus is fixed when there's only one allele in the population. So all individuals have the same allele. There's only one kind. There's only all individuals are exactly the same. That is a fixed locus. When you have more than one, you have at least two alleles, we say that the locus is variable. But there's something very important here. Usually, when we say that a locus is polymorphic, we do not mean by that that is variable. Variable means that we have at least two alleles. So there are individuals with two different alleles. Polymorphic means that the minor allele or the major allele have a frequency. For instance, usually we have as threshold for polymorphism 0.95 or 0.99. So polymorphic means that we have a major allele. A locus is polymorphic when its major allele has a frequencies that it's lower than 0.95 or 0.99. That depends on the criteria that we have. Okay, but not all locus are not all variable locus are polymorphic. So we have fixed loci with only one allele, variable loci with at least two alleles, and of those, some, usually not many, are polymorphic. Which ones? The ones in which the minor allele has enough frequency. Okay, it's not close to being fixed, because most of the variable loci we have seen are close to being fixed, but the polymorphic loci have more of the minor allele. Okay, that is important because in these loci that are variable but are not polymorphic, okay, these ones, in this case, the major allele is the most frequent allele. Well, that is by definition, but it's highly frequent. That means that almost all individuals in the population, but very few, will be homozygous for the major allele, okay, in these ones. But in the polymorphic ones, there will be more individuals with the minor allele. In the side frequency spectrum, what you have is here we have variable markers, so markers with more than two alleles. That is why we have a frequency in this case for the minor allele, but these ones will be not polymorphic. The polymorphic ones would be these, because if we choose as the polymorphic threshold this value here, these would be the ones that are polymorphic. Okay, now that we have talked a little bit about the definition of alleles and allele frequencies, let's talk about genotypes. In genetics, we have the difference between genotype and phenotype. Usually we talk that, we say that the phenotype is the observed characteristic, what we can measure, we can see in an individual, okay? The observed state of a trait, that is the phenotype. And the genotype is the hereditary makeup. In the case, if we are talking about markers, the set of alleles for one individual for a set of loci, that would be the genotype. So for each loci, which are the alleles for that individual, that is the genotype. If we have an ideal population with one, we are studying one locus, this locus has two alleles, and we have diploid individuals, we will have three possible genotypes. Two genotypes are homozygous. These are capital A, and lower a. And one genotype will be heterozygous, capital and lower A. We can calculate again the genotypic frequencies. The genotypic frequencies will be, we count how many individuals do we have for each kind of genotype, and we divide it by the total number of individuals. Those would be the genotypic frequencies. Of course, again, if we sum up all the genotypic frequencies, they will be 1 always. Why? Because every individual should have one of these genotypes. Okay, that is why when we sum up the genotypic frequencies, we have a sum of 1. If we have the frequencies for the genotypes, for instance, if we have genotype, the individuals, we know the genotypes, and we calculate the genotypic frequencies, we can always derive from those genotypic frequencies the allelic frequencies. That is very simple, because we have how many of each genotype do we have the allelic composition of each genotype, and we can sum that and we get the allelic frequency. Okay, this is a trivial thing to do. It's not difficult to do. However, going from the phenotypic the allelic frequencies to the genotypic frequencies, that is not trivial. Okay, in fact, in the general situation, in a general population, we cannot calculate the genotypic frequencies if we only have the allelic frequencies. That is not possible. Why? Because for two given allelic frequencies, we could have an infinite number of possibilities for the genotypic frequencies. Let's have an example. Imagine that we have one marker in a population and the allelic frequency is 0.5. Sometimes we have the allelic frequency, but we don't have the genotypic frequency. But in general, this will be useful because we can compare what we expect for a particular kind of ideal population, what we see. Okay, imagine that we have one marker and the allelic frequency is 0.5. In this case, Q would be 0.5 and P would be 0.5, of course, because they sum 1. Now, which would be the genotypic frequencies? We don't know, because we can have different possibilities. For instance, we could have a population in which we only have homozygous individuals. In this case, the genotypic frequency would be 0.5 for capital A, and 0.5 for lowercase a, and 0 for the heterozygous. And this population would have an allelic frequency of 0.5. But we have other possibilities, many other possibilities, infinite number of possibilities. But there's one in which we have, for instance, for the homozygous individuals, for the homozygous genotype, we have 0.25, and for the heterozygous 0.5. In this case, the allelic frequencies will also be 0.5. So we have different possibilities for the genotypic frequencies, even if we fix the allelic frequencies. So in the general case, we cannot derive their genotypic frequencies from the allelic frequencies. We can always do it because it's trivial the other way around. Okay, we can calculate always the allelic frequencies from the genotypic ones. There's a very special case in population genetics that we use all the time, that it's called Hardy-Weinberg equilibrium. And in the Hardy-Weinberg case, we do some assumptions, and in this case, you will see that there's a formula to calculate the genotypic frequencies from the allelic frequencies. But that is an ideal case, and well, there are many populations that are close to that case. That is one of the reasons why this case is used. But we will talk about that later. Another measure that is useful is the observed heterozygosity. That is, we count the number of genotypes that are heterozygous, and that would be the observed heterozygosity. You have to be very, very careful, because there's another term that is more common, and it's the expected heterozygosity. We will talk a little bit later about the expected heterozygosity. Okay, but the observed heterozygosity is just the number that we see. How many heterozygous individuals do we have? We divide it by the total number, that is the observed heterozygosity. And if you want, you can do an exercise. It's in the folder with all the presentations. You have this example of a population with 6 individuals in which you have 3 loci, and you can calculate all these allelic and genotypic frequencies. If you want to do it, you can check with the solution here. Okay, let's move on. Okay, now that we have defined allelic and genotypic frequencies, let's talk a little bit about diversity. Why is diversity important? Diversity is a crucial characteristic of the populations. Why? Because selection can only happen if there's diversity. If we can choose between different possibilities, it can be selection, but if we cannot select because everything is the same, then we cannot select. Okay, so diversity is critical. In breeding, for instance, diversity it's basically the material with which the breeder can work, and in natural selection also. So diversity is very, very important in every population, and there are populations that are very diverse and populations that are almost uniform. So we need measures that gives us an idea of the genetic diversity of the populations. We have populations that at the genetic level are highly variable, highly diverse, and populations that have very low variability. How could we quantify the diversity of the different populations or of different genomic regions? Well, we can think on different measures for this diversity, different indexes. For instance, in one index that we can use is how many polymorphic markers. Remember, polymorphic is we set a threshold and we calculate how many markers or how many loci do we have that are over that threshold for the major or the minor allele. Depends on which one you take as a reference. For instance, how many markers do we have in which the major allele is lower than 0.95? That would be the number of polymorphic markers, and we can calculate that number for each population and calculate the frequency, and that would be the proportion of the polymorphic loci. The more polymorphic loci that we have in a population, the more diverse is the population for that index. There's another index that it's sometimes used, it's not as used as the other one, but you can also use it. It's the mean number of alleles per loci. Imagine that we have a loci with only one allele. That locus would be fixed. There's only one, it's fixed. Which is the mean number of allele per loci if all loci in the population are fixed? If we don't have the diversity, that would be 1. Now, imagine that we have some loci that have 2 alleles. In that case, we could calculate how many loci we will have with 1 allele, how many with 2, and we could calculate the average. And for different populations, we might find that we have different measures. For instance, in a SNP, it's quite common to find that there are populations in which we have, for instance, 1.5 mean number of alleles. Okay, we have some alleles, some SNPs that are fixed with only 1 allele, and some other SNP with 2 alleles or with 3 alleles, and we calculate the mean. Okay. Of course, if we are talking about different kind of markers, like for instance microsatellites, the mean number of alleles for macrosatellites in a population could be very, very high. Okay, if we could have 10.5 as an average number of alleles for a marker. Okay, This is another measure of diversity. Then we could think, well, the number of heterozygotes in the population, it's indicative of the diversity, okay? That could be an idea. It's wrong, as we will see, but there's something in there. If we have a population with many heterozygotes, that it indicates that maybe this population is more diverse than the population with less heterozygous individuals. So heterozygosity, it's related with diversity, but there's a problem. If we have populations that are highly inbred, or there's some inbreeding, heterozygosity, the observed heterozygosity is not a very good indication of diversity, because we have two things mixed. How diverse is the population and how inbred is the population. Okay, we could have a population that is highly diverse, but it's very, very inbred, so we have no heterozygous individuals, so the observed heterozygosity would be 0. So observed heterozygosity is not an index that is used to calculate how diverse it's a population. But there's the other heterozygosity that I talked before. It's the expected heterozygosity. Expected, what do we mean by expected? Expected is if the population is in some conditions. For instance, one of the conditions is there's no inbreeding at all. There are other conditions that we will go talk next week. So if we have an ideal population that it's in the Hardy-Weinberg case, for instance, and one of those conditions is that there's no inbreeding, in that ideal population, given the allelic frequencies, we could calculate the number of homozygous individuals and heterozygous individuals that we expect. So we can have an indication of how many heterozygous individuals do we expect. In the case of two alleles, this number is 2pq. So, and another way of calculating this is 1 minus the allelic frequency p squared minus the allelic frequency q squared. In the general case, if we have more than two alleles, the expected heterozygosity, so the heterozygosity that we expect in a population that is in Hardy-Weinberg equilibrium would be 1 minus the allelic frequencies squared, the sum for all alleles. And you can calculate that for any population, even a highly inbred population, because you always have the allelic frequency. So one thing that you could think is, I have these allelic frequencies. Okay, if my population would be ideal and would comply with the requirements of the Hardy-Weinberg population, which would be the expected heterozygosity? You calculate that, and then with expected heterozygosity, you can compare between different populations, even if they have different levels of inbreeding. So this is the one of the most common indexes that we use to characterize the diversity of a population. It's called expected heterozygosity and also Nai's diversity. Okay, these two names are used for this measure, and it's very, very, very common. Okay, you will see that almost in any paper in which they talk about genetic diversity, this is one of the indexes that they calculate. It's I've been talking about markers and loci, but in general, a locus could be different things. Could be a marker. For instance, we could have two SNPs, two genetic markers, and the locus could be that, and we could calculate the diversities for each SNPs, and then we could do the average. That is a possibility, but there are other possibilities. For instance, we could consider a region with several markers, and we could consider the haplotypes, not the genotype of each marker, okay, but the haplotype. What do we mean by haplotype? Haplotype is if we have an individual, and imagine that we have diploid individuals, okay? Our species is diploid and we have diploid individuals, so each individual will have 2 homologous chromosomes. So if we have 3 markers, in general 3 loci, but imagine 3 markers, we will have the allele for 1 homologous chromosome for each marker and the allele for each homologous chromosome for the markers. The allele for the other homologous chromosome for all markers. The set of alleles in 1 homologous chromosome, that is an haplotype. Okay? In this case, for the locus A, the genotype is heterozygous. It's capital A, lower a. Okay? This is the allele, the genotype for the first marker. The genotype for the second marker, it's capital B. Capital B, it's homozygous. But if we consider the whole region and we talk about the haplotype, in this case, what we have is 2 haplotypes. The haplotype would be, one haplotype would be capital A, capital B, lower c, and the other haplotype would be lower a, capital B, capital C. Okay, that would be the genotype for the whole haplotype. So we can talk about genotypes for individual loci, okay, taking into account that in this case we are talking about markers, or we can talk about haplotype genotypes in general. And once that we are using haplotypes, we can use the alleles and the genotypes for the haplotypes, and we can calculate any index that we have been talking about. For instance, mean number of alleles over expected heterozygosity for the haplotypes. These haplotypes are relevant for different things. For instance, one important thing is that diversity is not just related with the genotypes of individual markers, but the haplotypes. We could have populations with the same alleles, but different diversities if we take into account the haplotypes. For instance, imagine that we are studying tomatoes and we have a population in which we have red big tomatoes and small yellow tomatoes. We have these two classes of tomatoes, okay? We have 2 different alleles for the red, if the trait depends only in one locus. We have red and yellow, and small and big, okay? 2 loci, and 2 alleles for each. We could calculate diversities, okay, for the individual loci, the individual locus. But we could have a population with the same loci and the same alleles with more diversity. We could have a population we have the red big tomatoes and the yellow small tomatoes, but also red small tomatoes and yellow and big tomatoes. Same genotypes for the individual marker, for the individual locus, but more diversity if we consider the combination of different locus, okay, if we consider the haplotypes. So the haplotypic diversity is interesting because it reflects a different kind of diversity, and it's a diversity that matters because selection selects individuals. The individuals reproduce or not reproduce, not the isolated markers. So what is selected is the combination of markers. So the haplotypes are selected, not the genotypes of individual markers. Okay, and also if we are trying to study the history of the populations, haplotypes we will see that are very, very interesting. The problem with haplotypes is that although they are very interesting, in many cases they are not used because it's difficult to genotype haplotypes. It's very easy to genotype with the technology that we have to genotype individual markers. For instance, imagine that we have an individual and we genotype one marker and another marker individually. For instance, with an SNP array, and we find that for the first marker we have the genotype capital A lower a, and the second marker it's also heterozygous. Okay, can we derive from this information that it's the information that we get from the genotype in the machine? So this is the observed data. Can we get the haplotypes? No, we can't, because there are two possibilities for the haplotypes. We could have that individual one with these genotypes. We could have that the haplotypes are capital A, capital B, and lowercase a, lower b, or alternatively we could have capital A, lower b, lower a, capital B. So from the individual genotypes, it's not we cannot directly calculate the haplotypes. We need either a technology capable of genotyping haplotypes, and that is not easy and usually not cheap, or we need a way in which we infer the haplotypes. There are ways of this is called phasing. For instance, there's a software that it's called Beagle2 that can infer that if you have many individuals in the populations, the assumption is that some individuals usually more or less will be homozygous, and in those it's trivial to derive the haplotypes, and with that you can build statistical models and infer the haplotypes. But it's not a trivial thing, and you need quite a lot of information in order to have a good inference. And finally, we also can have sequences. We could sequence, the individuals, and we could calculate diversities for sequences. Usually when we have sequences, the most used measure it's called nucleotide diversity, usually represented as pi, and the nucleotide diversity is the equivalent of expected heterozygosity. It's very similar, but for sequences. And I have prepared an exercise that I recommend you to do. You can go here, and in this sheet you have diversity indexes exercise. You have 3 populations with genotypes for 5 SNPs, and you could calculate all these indexes. If you want, the solution for the exercise is here, so you can check if you have your index is calculated as expected. Finally, we have calculated different indexes. Do all these different indexes tell us the same story about how diverse are the populations compared with each other? No. Different indexes, as we will see, give you different ideas about the diversity. So the diversity is, in general, it's not completely a term that it's well defined, because one population can be more diverse than another if you take into account one index or if you take into account another index, and that would mean different things for the breeder or for people that study history or different things. We are going to talk about this a little bit with an example, because the general case is very complex. But we are going to see an example in tomato, because we have studied tomato quite a bit. I am going to show you diversities calculated in tomato population, and we are going to see how different measures compare. One measure that we have seen is the allele frequency spectrum. Here you have the allele frequency spectrum for 3 populations in tomato. We can compare, for instance, modern tomato and vintage tomatoes. And as you see here, modern tomatoes are more diverse than vintage tomatoes according to this measure, because in modern tomatoes there are more markers with with lower major allele frequencies. In this case, we are representing the major allele frequency histogram. So in this case, the vintage tomato has lower diversity than the modern tomato. Which one will have less polymorphic markers? Well, if we set the number of polymorphic markers as the markers that have a major allele frequency lower than 0.95, the population with more markers, more polymorphic markers, would be the modern population, okay, not the vintage one. In another paper, we studied the populations in America. We defined, using PCAs, different populations, and within this population different subpopulations, and we have calculated the measure, for instance, number of polymorphic markers. And you see here that we have populations that have more diversity according to this measure, like for instance this Pipin lelifolium from Peru and Ecuador have quite a bit of diversity compared, for instance, with the diversity that we find in Cerasiformi in Mesoamerica. Also, the Cerasiformi SLC, it's CS from Cerasiformi in Peru and Ecuador, they have also quite a bit of diversity compared with Mesoamerica. We also can calculate the number of variants, the number of, for instance, SNPs, but in this case it's SNPs and small insertional deletions, and the result is more or less similar. There's a difference in this population. This population is a hybrid between the wild and the cultivated species, and in this case we have more markers but than in Cerasiformi from Ecuador, but less polymorphic markers. That would mean that in this population we have many markers but many of them have allele frequencies below the 95% threshold that we have set, so they are not polymorphic. If we have gene flow of admixtures, these different indexes that we have been talking about could be quite different between populations. For instance, we have calculated now the number of polymorphic variants and number of variants in general. But let's see what happens if we calculate the mean number of different haplotypes. So how many alleles do we have? But take into account haplotypes. That means that we take segments of the genome, we divide the genome into windows, and we calculate the haplotypes for each window. And we calculate for each population how many alleles do we have in each window, and we can calculate the mean for all windows, and this is what we get. If you remember, when we calculated the number of polymorphic markers, Cerasiformi from Ecuador had very high diversity. You see, here. But when we calculate the mean number of different haplotypes, it's quite similar to Cerasiformi from Mesoamerica. So there's a stark contrast between this measure and the previous measure. You see, here, if we calculate number of polymorphic markers, the number of polymorphic markers in Cerasiformi Ecuador is much higher than the number of polymorphic markers in Mesoamerica. But when we calculate the number of different haplotypes, it's very similar between these two populations. So according to this measure, Ecuador looks like a highly diverse population, but according to this measure, Ecuador looks like a population that is not so special compared with Mesoamerica. So it depends on the measure, we get different results. And as I have said, introgressions and gene flow can have important differences. For instance, why introgressions are so affect so greatly these diversity measures? What is an introgression? An introgression is that we have an haplotype from one species or one population, and we put it in another one. So we have in the receiving population, now we have an haplotype that is quite different than the haplotype in the rest of the haplotypes. Imagine that we have a species in which we have only one haplotype in the previous haplotype in the region, and now we have an introgression and we have an haplotype that is quite distinct. In this case, the number of variants, the number of markers would be very, very, very high. Okay, we have 1, 2, 3, 4, 5, 6, 7, 8 variants just with one haplotype. Only one haplotype, sorry, two haplotypes, the original one and the new one, but 8 markers. Well, technically it's called 8 variants usually. In a more standard population without introgressions, in which we have more diversity, we have only 2 markers, but these 2 markers are polymorphic. In this case, we have many variants, so if we calculate the number of variants, we will calculate there are many variants, but no polymorphic variants. And in this case, we will have fewer variants, but the variants are polymorphic. So, as you see, the introgressions bias some of these diversity measures. So, by calculating indexes for the different kind of measures that we have explained, you can have an idea of what is going on in the structure of the variation. Also, you have to take into account that for many of these measures, the number of individuals matter. So if you have populations with many individuals and with fewer individuals, you have to be very, very careful when you compare these diversity measures. For instance, the number of variants. Well, the number of variants depends on the number of individuals that you analyze. This is a rarefaction analysis. Rarefaction analysis is an analysis in which you have a number of individuals. For instance, in this case, we have 500 vintage European tomatoes, but you analyze first, for instance, 10 individuals, 11 individuals, 12, 13. You go on and adding individuals, and you calculate the index that you are studying for the different number of individuals chosen at random from the population. And as you can see, the number of markers, the number of variants, goes up with the number of individuals. Why? Because as we have explained, most variants are in a very low, has a very low allelic frequency. So if you add a new individual, you will find markers that weren't previously found in the population because they are in a very low frequency and they are almost in very few individuals, in the individuals that you just added and maybe in another one or few, or maybe just in that one. So if you keep on adding individuals, you will find that this measure goes higher and higher and higher. Also, the expected heterozygosity goes down with the number of individuals usually. Why? Why? Because if you add more individuals, you have more markers. If you have more markers, the new markers that you have added will be markers that are usually that will have usually a lower allelic frequency. Since the expected heterozygosity is calculated from the mean of all markers, if you are more individuals, you are more markers with lower diversities, and the mean goes down. Okay, you have to take that into account when you are working with expected heterozygosity. One alternative is use a modified expected heterozygosity that it's called unbiased expected heterozygosity, that it's calculated, its definition, it's precisely established to avoid this problem. So the unbiased expected heterozygosity does not depend so much with the number of individuals, but it also depends a little bit about with the number of individuals, so you have to be careful. The number of polymorphic variants is more robust than these previous measures because at the very beginning, when you have very few individuals, it depends just by chance of which markers you are seeing. But when you add more individuals, it reaches a point in which this measure is stable. Why? Because even if you are more individuals with more markers that are in low frequency, these markers that are in low frequency are not polymorphic. So the new markers that you discover by adding more individuals are not polymorphic. So this measure, the number, not the proportion, but the number of polymorphic markers does not go down. And that's all for the example of the tomato. Yes. Okay, let's move on. We have been talking about diversities, but another very important measure, it's a measure related with distances. So in diversity we have one population and we calculate how diverse it is. It's very diverse or it has a very small diversity, okay? But now we are interested in comparing different populations. How different are they? There are many measures of distance between individuals. I am not going to go into the details of how these distances differ. They are, I think, more or less similar. Depending on the kind of markers, similar that at the end you get more or less the same ideas, although there are technical differences between them, okay? It depends on the marker that you have, if they are dominant markers or things like that. You could choose one measure over the other, but if you have SNPs, one commonly used measure, it's called Cosman measure. Here you have the reference for this measure. I am not so interested in this case of talking about the distances between individuals, but the problem of calculating distances between populations. When you have populations, you don't have just one individual in one population and one individual in the other population, okay? you have more than one. So you have many possible distances between the populations. And one thing that you could think, well, I could calculate all these possible distances and I could calculate the mean. And that would be the distance between the population. That would be quite misleading, or because you would forget something. You would ignore that you also have distances within populations, okay? They are related with the diversity of the populations. So usually when we talk about genetic distances between populations, we take into account the distances within the population, the diversity within the population. Why? Because two populations with the same distance between them, the same mean distance between them, could be very different or could be very similar. In this case, we have two populations that have the same mean distance between them, but in the first case, these two populations are very different. They do not share a lot of diversity, but in this case, the populations are quite similar. They share a lot of diversity. That is why it is important to take into account the distance within populations when you calculate the distances between populations. One way to take this into account is to calculate two different kinds of diversities. You could calculate the diversity for each population and then you calculate the mean diversity. That gives you an idea of how diverse in general are the populations, and then you calculate the diversity of all together, okay, the total diversity. If both populations are very different, the total diversity will be much higher than the mean diversity for each population. If the two populations are very close, the total diversity would be very similar to the mean diversity within populations. Okay, that's the idea of most of the distances between populations. One very popular measure is FST. There are others that we will see, but this is one very popular one. FST, it's you calculate the total diversity by taking into account the individuals from all the populations. Then you calculate also the diversity for each population isolated, and you calculate the mean for diversity. And it's FST total diversity minus mean diversity for every population divided by total diversity. This is good genetic distance between populations when used for two populations, okay? Imagine that we have two populations that are completely different. They do not share any diversity, okay? The polymorphic markers in one population are, the markers in one population are fixed in the other and the other way around. So in this case, if we calculate the total amount of diversity taking into account individuals for both populations, we will have a very high diversity. But if we calculate the diversity within one and the other, we have, and we do the mean, the diversity within both populations is low. So if you go to the formula, the total diversity would be very high, the mean diversity for each population very low, so something very big minus something very small divided by this big something, that gives you 1. So FST will be 1. If in the other case most of the diversity is shared, then FST would be almost 0, close to 0. So FST can go from 0 to 1. 0 would be these two populations are almost the same in terms of diversity, and one, these two populations are very different. Okay, this is the example that I have been talking about. And you can have all the spectrum between 0 and 1. In human populations, FST is quite low. It depends on the marker that you use and the study, but in general it's quite low in every estimate of the FST for all human populations. That means that most of the diversity is shared within populations. So you have within a population individuals that are so far away as almost individuals with other populations. You can also calculate FST with any diversity, for instance with pi. That is also used when you have sequences. So you can calculate FST from sequences, not just from molecular markers. There are many ways of defining and estimating FST. Okay, this is a very complex topic and I am not an expert, but it's important to have the idea what FST means. How to calculate it can be quite involved depending on the author. There are other measures. For instance, another measure that is sometimes used is the Ney distance between populations, okay, or the Ney standard measure. But there are many other. For instance, you have Cavalli's Forza. There are many distances, okay. Neys have even different distances. But the general idea is that you have to take into account the diversity within populations and between populations. And you have a problem. If the diversity between the populations are very different, then these measures will be quite misleading, okay? In this case, these two populations do not share any diversity, but FST would be low. So FST or almost any other measure that is used usually to calculate distances between populations work quite well if the diversities are similar, but if the diversities are very different, they can be quite misleading. You have to take that into account. A single number cannot reflect the distance between the populations very well. Okay, and just to finish, we are going to talk about another measure that is used to characterize a population that is called linkage disequilibrium. In here, we are talking about the relationship between different loci. Let's take a look at this example. In here, we have two different populations. In the first population, we have that the genotypes for both loci are highly correlated. If we know the genotype for one locus, we know the genotype for the other locus, okay? The correlation is very high. In the other example, the information about the genotype of one locus does not say anything about the genotype of the other locus. So we have measures that tell us about this correlation. It's usually in genetics we do not talk about correlation between loci, we talk about disequilibrium. Two loci that are completely correlated, we say that are in disequilibrium, and two loci that are completely correlated, we say that are in equilibrium. Why are loci in equilibrium? Ultimately, they are in equilibrium or disequilibrium because we have linkage. Okay, so since we have linkage, there's a tendency that the markers that are close will be, the genotypes will be highly correlated, and when we go far away, the genotypes will be less and less correlated. Of what depends this level of correlation? Well, it depends on the number of recombinations that have happened in the past. Okay, so, and this number of correlations depends on several things. One is the genetic distance, but also about the genetic composition of the population and the diversity of the population. So genetic distance, it's a very important thing for linkage disequilibrium, but it also depends on the diversity and the history of the population in general. When we have linkage equilibrium, what we have is a statistical independence between the genotypes of the two loci. That means that if we have information about one loci, we don't know anything about the other locus. In this case, and only in this case, we can estimate the expected frequencies for the haplotype by multiplying the allelic frequencies of the two loci. Okay? Only in this case in which we have independence. So one way to measure this disequilibrium is we calculate what would we expect if we had this equilibrium. We calculate the frequency of the haplotypes, the real ones, and we see the difference. Okay, this is one way in which linkage disequilibrium is calculated. This measure is usually normalized, so you will see in some papers that they talk about D prime. But a more common measure is R squared. R squared is just the correlation between the loci, and here you have the definition of R squared. When you are in complete linkage disequilibrium, the R squared is 1. The correlation is complete, it's R squared 1. And when you are in complete equilibrium, that would be R squared 0. No linkage disequilibrium, it's R squared 0. A very common way of representing linkage disequilibrium between different markers is to represent the matrix, well, half of the matrix, because the matrix will be symmetric, in this way. You put the markers in a line, and you put the correlations between the lines that would join the two markers. For instance, this 77, it's a correlation between this marker and this marker. Okay, and what you find in a genome, if you represent the linkage disequilibrium between the different pairs of markers in the genome, is that you find blocks of linkage disequilibrium. There are blocks in which the information of all markers are highly correlated, and then you jump to a new block and you have that the information of the markers are correlated within the markers in the block, but not between the markers in the block and markers in other blocks. This is quite common. Okay, that depends on where the recombinations have happened in the past. Okay, but and these blocks are usually used to select markers to put in genotypic arrays for genotyping, because you don't need to put every marker in the block because they are highly redundant. So they tend to put some markers within the block, but they make sure that we represent the different blocks. So one factor that affects linkage disequilibrium, of course, it's the distance. Linkage disequilibrium, when you are working with markers that are very close in the genome, usually it's quite high. and when you go far away, usually the linkage disequilibrium goes down. But the rate at which the linkage disequilibrium goes down with the distance depends a lot of different species. Okay, for instance, you have here an example in melon and in sorghum, and in melon the linkage disequilibrium you have a mean R square 0.2 when you are in 80 kilobases, and in sorghum this same number is reached much earlier. So the linkage disequilibrium in sorghum is lower, for instance, than in melon, or in tomato it's much higher than in both, especially in cultivated tomatoes. So the degree at which linkage disequilibrium goes down with distance depends on the history of the population. So depends on the population. And finally, I am not going to talk about statistics a lot, but if you really want to understand linkage disequilibrium, I recommend you to think about probability and independence. I am not going to go into detail. I am not a statistician, but I recommend you to, if you are interested in linkage disequilibrium, to read about probability and independence. In probability, they say that two events are independent if one event, the frequency of one event does not influence the other. Okay, in that case, we have two independent events. For instance, if we throw two dice, then these two, the outcome of each throw, it's independent from the outcome of the other throw. In that case, we can use the rule of the product, and that is what we have used to calculate the expected frequency for the haplotypes if there were no no relationship between the genotypes of two markers. Okay, we have used this, the rule of the product. The rule of the product means that the probability of two events happen is just the frequency of one event happening multiplied by the frequency of the other event happening. But this is only true if they are independent. There are many events that are not independent. For instance, the event of testing positive for a COVID test is not independent. Better said it. The event of you being infected with COVID is not independent of you having had a positive test. Okay, these two events are clearly not independent. When you have events that are not independent, then you cannot use the naive rule, the multiplication rule, to calculate when the two events happen both. Then you have to multiply the probability of the first event multiplied by the probability of the second event given the other event. Okay, this it's a conditional probability. That means the probability of B happening given that we know that A has happened. Okay, these are conditional probabilities. And related with that, there's a very important idea that is not related with linkage disequilibrium directly, but it's a very important idea in general in science, and I recommend you to read about this. It's called the prosecutor's fallacy or the base rate fallacy. For instance, if you hear the sentence, the suspect in a murder case, for instance, has O+ blood and dark curly hair, and O plus blood and dark early hairs was found in the scene of the crime, so the suspect is guilty. If you hear that and you think that it's correct, then you are committing the prosecutor's fallacy. Fallacy, it's that it's a logical mistake. Another thing that will be the same kind of fallacy is if you don't have COVID-19, in the case that you don't have COVID-19, the probability of having a false positive in a test is 1 in 100. So if you have had a positive test, the probability of not having the disease is 1 in 100. If you think that this is true, you are committing what is called the base rate fallacy. It's the same kind of fallacy. It has two names. Okay. Basically, what you are doing is that you are thinking that the probability of A given B is the same as the probability of B given A. I am not going to go into there, but this is a very natural thing to think, but it's wrong. Very, 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 very wrong. Okay. These probabilities are not related like that. They are related with something that is called the Bayes theorem. And I recommend you to read about this because it's important in science, especially if you work with p-values. You have to take that into account. Well, almost if you do any kind of statistical inference, it's important. Okay, and that's it for today. Thank you very much. We will see you next class.